A warm welcome to one and all present over here. Dear friends, myself Dr. Rashmi Joshi Sawalkar, Assistant Professor, Department of Zoology, Shri RLT College of Science, Akola. Today, here is our sixth lecture on Unit 2nd, and the topic is contraction of muscles. So, let's start. Friends, in the previous lecture, that is in fifth lecture, we completely studied the topic sliding filament theory, and as there is a gap between these two lectures, I want to recall you that what is the sliding filament theory. So let's see some points about the theory. During muscle contraction, myosin cross bridges pull on the thin filaments, causing them to slide inward towards the H zone. Z discs come towards each other and the sarcomere shortens, but the thick and thin filaments do not change in length. The sliding of filaments and shortening of sarcomeres causes the shortening of the whole muscle fiber and ultimately the entire muscle. When a nerve impulse, that is nerve action potential, reaches an axon terminal, the synaptic vesicles of the terminal release the chemical acetylcholine, which is a neurotransmitter, which ultimately initiates a muscle action potential in the muscle fiber, sarcolemma, then travels into transverse tubules and causes some of its scored calcium dipositive to be released into the sarcoplasm. The muscle action potential releases calcium ions that combines with troponin, causing it to pull on tropomyosin to change its orientation, thus exposing myosin binding sites on actin. The immediate direct source of energy for muscle contraction is ATP. ATPase splits ATP into ADP and inorganic phosphate, and the released energy activates myosin cross bridges. Sliding filament mechanism. Sliding filament mechanism. Activated cross bridges attach to actin and a change in the orientation of the cross bridges occurs. This is called as power stroke. This movement results in the sliding of thin filaments. Once the power stroke is complete, ATP again combines with the ATP binding sites on the myosin cross bridges. As ATP binds, the myosin head detaches from actin and the cycle may be reinitiated repeatedly. So, friends, this repeated detaches and attachment of the head part of the actin filament helps in the contraction and shrinking of muscles, and the exact opposite process is the relaxation. Now, relaxation is brought about when ACH is broken down by the enzyme acetylcholinesterase and Ca positive is moved from the sarcoplasm back into the sarcoplasmic reticulum. Calcium removal is accomplished by active transport pumps and a calcium binding protein called calciquestrine. This was all about the sliding filament theory. I think you all got it that what is this sliding filament theory, as the next topic of muscle contraction is completely related with this topic. So now let's see the other title, muscle tone. What is this muscle tone? A sustained partial contraction of portions of a relaxed skeletal muscle results in a firmness known as muscle tone. At any given moment, a few muscle fibers within a muscle are contracted while the most are relaxed. This small amount of contraction is essential to maintain the posture. See, when the complete muscle is relaxed and some part of that muscle is somewhat firm or contract, so that small part is called as muscle tone. Okay, now this muscle tone may be studied in further in two parts: hypotonia and hypertonia. Then, what is hypotonia? Decreased or lost muscle tone, such muscles are said to be flaccid. And hypertonia, increased muscles tone, such muscles are said to be either spastic or rigid. So, these are the terminologies which are used for the muscle tone: hypotonia and hypertonia. Now, what is rigor mortis? This is again the new term in our unit, rigor mortis. On this topic, many times 4 marks short note is asked in the examination. So let's see what is rigor mortis. Rigor mortis is a state of muscular rigidity following death. See, it causes death. Rigor mortis is a state of muscular rigidity that muscles get rigid, and this rigidity causes death also. It results from a lack of ATP to split the myosin actin cross bridges. At the time of contraction and relaxation, we studied in the previous slides. Recall the topic that at the time of contraction and relaxation, myosin actin cross bridges, ATP is required, and a few hours after a person or animal dies, the joints of the body stiffen and become locked in place. This stiffening is called as rigor mortis. Now, depending on temperature and other conditions, rigor mortis lasts approximately 72 hours. See, after death, in some animals or in some persons, the rigor mortis lasts approximately for 72 hours, because it depends on the climatic conditions or temperature, and other conditions too are responsible for rigor mortis to last for 72 hours. The muscles are unable to relax, so the joints become fixed in place. The muscle cells become more permeable to calcium ions. Living muscle cells expend energy to transport calcium ions to the outside of the cells. The calcium ions that flow into the muscle cells promote the cross bridge attachment between actin and myosin. The muscle fibers ratchet shorter and shorter until they are fully contracted, or as long as acetylcholine and ATP are present. However, muscles need ATP in order to release from a contracted state. It is used to pump the calcium out of the cells so the fibers can unlatch from each other. As ATP is released, and it is needed by the muscle in order to release from a contracted state. When ATP is released, the contracted muscles get relaxed. So this important function is done by this ATP molecule. ATP reserves are quickly exhausted from the muscle contraction and other cellular processes. This means that the actin and myosin fibers will remain linked until the muscles themselves start to decompose. This is rigor mortis. Now, rigor mortis can be used to help estimate time of death. The onset of rigor mortis may range from 10 minutes to several hours, depending on factors including temperature. Now, rapid cooling of the body can inhibit rigor mortis, but it reoccurs upon thawing. Thawing is a new term for you all. I want to explain what is thawing. That anything or any chemical or anything that is placed in very lowest temperature, that is below minus 100 degrees centigrade, and uh, to bring it at the normal temperature, the steps are followed by slowly increasing the temperature, and this slow increase in temperature is known as thawing. Maximum stiffness is reached around 12 to 24 hours 12 to 24 hours 12 to 24 hours post-mortem. Facial muscles are affected first with the rigor, then spreading to other parts of the body. The joints are stiff for 1 to 3 days, but after this time, general tissue decay and leaking of lysosomal intracellular digestive enzymes will cause the muscles to relax permanently. Now, let's see what is the muscle metabolism. On demand, skeletal muscle fibers can step up ATP production as the requirement of the body or after getting heavy exercise or after doing heavy exercise, the body has some demand of ATP molecules or energy. So skeletal muscle fibers can step up the ATP production. Creatine phosphate and ATP constitute the phosphagen system. This energy system can power maximum muscle contraction for about 15 seconds and is used for maximal short bursts of energy, such as those used by athletes in the 100-meter dash. The partial catabolism of glucose to generate ATP occurs in the glycogen lactic acid system. This is an anaerobic system. See, the partial catabolism of glucose, not complete. The partial catabolism of glucose, that is breakdown of glucose to generate ATP. In the generation of ATP occurs in the glycogen lactic acid system. This is an anaerobic process. Now, the system can provide energy for approximately 30 to 40 seconds. This type of energy or ATP generated provides approximately 30 to 40 seconds of energy of maximal muscle activity. This would be useful in the 30-meter dash. Muscular activity requiring more than 30 seconds, but if the muscle activity requires more than 30 seconds of maximal energy, then the utilizes aerobic system. Then the oxygen that utilizes the aerobic system. This system of ATP production involves the complete oxidation of glucose via cellular respiration or biological oxidation. Muscle tissue has two sources of oxygen, one from the muscle fibers from the blood and oxygen that is released by myoglobin from the inside of the muscle tissue. It means muscle tissues receives oxygen from the bloodstream and from the muscle tissue itself. The aerobic system will provide enough ATP for prolonged activity so long as sufficient oxygen and nutrients are available to the muscles. Now, let's sum up our topic and let's see what is muscle fatigue. Muscle fatigue, the inability of muscle to maintain its strength of contraction or tension. See, friends, muscle fatigue is a state in a heavy exercise. When the muscles are in heavy exercise, they require more and more ATP molecules or energy. When the energy, when the energy they didn't receive the energy in the ample amount, this stage is called as muscle fatigue. So it occurs when a muscle cannot produce enough ATP to meet its need. At this stage, the muscle fatigue took place. So friends, today in the sixth lecture, we studied the muscle contraction, rigor mortis, the term rigor mortis, then muscle fatigue, and we revised the fifth lecture again in this lecture too, and it was the sliding filament theory. So I think now you completely understood the topic, how muscle contraction takes place, how the muscle fatigue and what is rigor mortis and all. So friends, do revise the lectures we are giving to you and study well, as there is your MCQ test. There will be your MCQ test soon and it will be informed about the date and schedule. Okay, thank you, thank you very much.